All right. I think we will get started, and hopefully Pontiano will join us in a moment. I'm Mitchell Warren, the director of AVAC, and delighted to welcome many great friends and colleagues to this call. It is mid-May, and as we have done for now 24 years, recognize May 18th as HIV Vaccine Awareness Day, and we decided it was helpful. And can you hear me? I see Barney. Levi, can you just confirm that I can be heard?
Yeah, I can hear you.
Okay, great. And here's Pontiano. Okay. And hopefully Barney, you can, oh, now Barney can hear. Fabulous. So now we're ready. As I was saying, next week, Tuesday, May 18th, will be 24 years since then President Bill Clinton gave a commencement address at Morgan State University, and quite a remarkable speech, which you can find online, both the text and the video, where he talks about the 21st century being a century of science. And chief amongst that was to accelerate the development of an HIV vaccine. He'd hoped that there would be progress in a decade, and that's been a common refrain. And clearly 24 years later, we are still in that search. But I think we all recognize that the 21st century, and certainly the last year, has been a year of science for sure, and delighted to welcome advocates, researchers, product developers onto this call to reflect a bit on the experiences of the last year, the experiences of the last 24 years of HIV vaccine R&D, and what we need to be doing more of and differently going forward. To give you a quick sense of the run of show, we are going to have a quick introduction of where things stand in 2021 from my friend and colleague Daisy Oya from AVAC, and then we're going to turn to the bulk of the call to a really exciting conversation with five remarkable advocates for HIV vaccines, and a couple of them also do a little research on the side, but all five being remarkable champions and advocates for advancing science and working towards ending epidemics. I want to encourage people to use the Q&A feature in your Zoom interface that I'm sure you're all familiar with, and we'll get to those as quickly as we can as Daisy and I moderate this great panel. Feel free to begin to put your questions in there from now, and we will get started in a bit with the questions. But first, I'm going to turn it over to Daisy, our communications advisor based in Nairobi, Kenya, and a critical part of the AVAC research engagement team, to give us a quick overview of where we are with vaccines circa May 2021. So over to you, Daisy.
Thank you so much, Mitchell. And I'm going to get right to it. This is going to be a very brief presentation on the just some basic, just to get our juices flowing, and a status update of where we are in terms of the search for a safe, efficacious HIV vaccine. So the first thing that I think we wanted to put out there is that vaccines work. They're one of the most powerful public health tools that are available. And many of us cannot remember hearing of a case of smallpox, let alone measles and poliomyelitis, right? Because these have become exceedingly rare. Some of them have been actually eradicated because of vaccines. And we've seen from the COVID-19 experience that we have been clamoring for a vaccine, and now we found not one, but several vaccines. And they will be essential to ending the COVID epidemic, and to end the HIV public health challenge. We will need an HIV vaccine. Next, please. This is a little infographic showing how long it takes to develop a vaccine. COVID is there on the second bar, which is completely unprecedented. You can see that typhoid took 105 years. People were born and died before they found a typhoid vaccine. 47 years for polio, and so on. So HIV research is still ongoing, 32 years, and we hope with the developments that are happening very quickly now, perhaps it's in sight. But let's keep going, yeah. So this, I think this is not a message, it's a message, of course, for everyone, but I think the people on the call actually understand very well why an HIV vaccine is needed. The little chart you see on the right-hand side there is how far we are from the UNAIDS targets for 2020. We are quite far from reaching them. Currently, well, as of 2019, based on the 2020 UNAIDS report, about 1.7 people became newly infected with HIV. So we are very, very far to reach the 500,000 target by 2020. Well, 2020 has passed and we are still at 1 million plus. So that's why an HIV vaccine is still very desperately needed, as well as other prevention options, I might add. You can keep going. HIV vaccine development is quite challenging. I know people are talking about the COVID vaccine experience, how fast we've made a vaccine, but this is just to point out that the COVID, SARS-CoV-2 virus and the HIV virus are quite different. No one has ever eliminated HIV with their own immune system vis-a-vis COVID, where people have become infected and many of them have recovered. So there's no roadmap to protection in terms of developing an HIV vaccine. And of course, the variability of HIV, it mutates continuously. There are so many, subtypes and clades and so on around the world. So to make a vaccine becomes that much more challenging. And of course, HIV attaches itself and kills the very cells that the body uses to defend itself against disease. So it gets into the CD4 cells and starts multiplying in there. And then of course, transmission of HIV is also complex. There's vertical transmission, there's sexual transmission, there is blood-borne transmission through injection needles as well as transfusions, and so forth. So it just makes it much more challenging. So the researchers that are on the call can talk more about this, yeah, because they are dealing with it on a daily basis. Yeah, keep going, Levi. Yeah, so I don't want to get it very sciencey because we have the scientists on the call, but this is just a snapshot of some of the types of immune responses that vaccines seek to elicit, by a vaccine. So we have the T cell mediated response, your killer T cells and your helper T cells, neutralizing antibody response, non-neutralizing antibody response, as well as combination responses, which various vaccines are designed to trigger in the body so that it can protect you from acquiring HIV when you encounter it. This is not a slide that I expect anyone to actually read because it's quite small. The graphics are quite small, but let's just say that the HIV pipeline is quite full. But as you can see on the left-hand side with preclinical and phase 1 and 2, that's where a lot of work is happening. In phase 2B to 3. This means efficacy trials. We currently have just about 3 trials, and so this is a pipeline that needs to be supported, very strongly so that we can move more candidates into efficacy so that eventually we can get an HIV vaccine. Yeah. So the current prevention trials are there. I think we can, these are prevention trials of all sorts of prevention options. So we have rings, which are already going into open label, not even open label, but demonstration projects. We have oral prep trials, including monthly prep pills. We have the Kabele, which has been now approved and hopefully will start getting rolled out. We have also injectable PrEP, lenacapavir by Gilead. We have the AMP trial, which released results just recently. Very, very nuanced results, if I may add. Yeah, and then down there we see the vaccine trials that are currently happening. Yeah, we can move. So I mean, 2021 is unprecedented in terms of what has happened around vaccines. We have now people are talking about antibodies just, like in the street, right? And vaccines especially. And, we have millions of people who have been newly vaccinated with new vaccines. So we are learning a lot of lessons from COVID for HIV vaccines. And the first one is that sufficient funding and funding from diverse sources will be critical. That's what we saw in COVID. Governments, pharma, nonprofits coming together to pull together money and push the research forward. Another very enabling factor was the coordination and collaboration. The data was open. The labs were working from the US to South Africa to China and so on. And then support for research innovation so that no good vaccine approach is ignored or is underfunded. And of course, the new trial designs that we saw, there were huge trials adapted, as the pandemic unfolded. This is what I think we need also for HIV vaccines. Political commitment and urgency is also a critical part, as well as placing communities at the center of vaccine research. This goes without saying because communities are the key, not only as participants, as well as people who can educate others about vaccines, address misinformation, build vaccine confidence, and so forth. And then, of course, the last one there is planning for success so that you don't wait until you get a perfect vaccine before figuring out, where is it going to be manufactured, who's going to be, delivering it, how is it going to be delivered, and where is it going to be delivered, most importantly, because equitable access is key, because as long as anyone is still vulnerable to an infection, I think we all are. It's a global world, and so equitable access has been a challenge for COVID vaccines, and we can learn lessons there for the day we have an HIV vaccine. Yeah, yeah. So that's the end of my short presentation, and you'll find key resources on the AVAC website. We have actually a page with all these resources, PowerPoints and messages, these key messages, links to various happenings around HIV vaccines, as well as some of the op-eds and interesting publications that have been published around this day. Thank you so much.
Great, Daisy. Thank you so very, very much for that terrific overview, and an extended version of that slide deck and the materials that Daisy mentioned are later today available at avac.org/HVAD, and so we would love for people to use those if they are helpful, but they were certainly helpful, Daisy, in setting the stage for this conversation. I see a couple of questions already coming in. Want to encourage more of them to come as Daisy and I get the joy of interacting with 5 terrific friends and colleagues. I think the easiest way to jump into the conversation, and maybe we'll introduce each of them in turn, is to maybe have them each take a chance at an introductory question in terms of how COVID vaccine research over the last year has influenced your work in HIV vaccines as advocates and as researchers, just to help set the stage. And I think for ease, we'll do it maybe alphabetically by first name. So I'll start first, Barney, with you, Barney Graham, who is, I think, likely better known to the world now than perhaps he was a year ago, is at the Vaccine Research Center at the NIH. And in fact, going back to Bill Clinton's speech in 1997, in fact, one of the great outgrowths of that speech was the creation of the Vaccine Research Center, which has pioneered work in HIV and in many other disease areas, including in COVID. So Barney, why don't we start with you? How has COVID vaccine R&D influenced you?
Well, thank you, Mitchell. It's a pleasure for me to be here with you today. Honestly, I think it's gone the other way around. My 40-year experience with HIV and my 35 or 6-year experience in working in research to be a viral immunologist, all of those threads of experience and learnings about both immunology and new technologies that have been developed in part through my connection with the Vaccine Research Center, which was the announcement that started HVAD back in 1997. It's really all come together to inform how to respond to this new virus that, as Daisy said, doesn't have the same kind of biological difficulties in making a preventive vaccine. And so all these things that we've learned from HIV have really informed and been at the basis for how the world has responded to this coronavirus.
Outstanding. I know we'll come back to that and your work in all of these areas. Let me next turn, we'll go from outside of Washington, D.C. to Harare, to you, Definite Namo, a leading advocate and social scientist based at Pangaea Zimbabwe AIDS Trust, has worked across a range of HIV prevention activities. And so, Def, what's the view from Harare?
Thank you, Mitchell, for having me here. A really exciting conversation. For me, the COVID vaccine has been met with mixed feelings, really, and still battling to understand how we still have a lot to learn in terms of vaccines, understanding that not all vaccines are the same, not all viruses are the same. Some take more time to develop, some take less time. But for me, the critical thing that I'll leave you with at this moment is, for me, the battle is how to communicate this to the communities we serve, that we've spent about 3 decades looking for an HIV vaccine and still going. With COVID, it has just taken a year. For me, it's still a difficult position to try and communicate that to the communities we serve, and really excited to understand this more through this webinar today. Thank you.
Great, Def. Thank you so very much. Delighted next to turn to Linda Gale Becker. She was once introduced just a few months ago as she reached stardom when you're known with a single name, so LGB, who leads so much research and implementation in South Africa and globally, a past president of the International AIDS Society and the deputy director of the Desmond Tutu Health Foundation based in Cape Town. LGB, COVID and HIV and the experience for you in the last year.
Yeah, so Mitchell, I would agree on definitely with Barney that years of being in HIV stood us in extraordinary good stead for the last couple of years. But doing it the other way around, the thought that comes to my head is go big or go home. It has expanded my horizons in ways I would not have known were possible. So everything has been orders of magnitude bigger, faster, and I think has blown away a lot of the myths we had that regulatory would take 6 months, that you moved from one phase to the next phase, maybe in a year type, time frame. And that, you enrolled a couple of tens of 20s, maybe, of people into your trial sites. That has all been blown out of the water in as much as I think there was a incredible global thrust to say, we will find a vaccine. And, thank you, Barney, for your role in that, in terms of a COVID vaccine, and we will do it in this particular time frame. And, we did it. The royal we made that happen. And as I say, I think it's just, for the world will never be the same again, because the genie's out of the bottle. We now know what we can do when we put our minds and our hearts to it. And, not underestimating the complexity of the science. It is a different virus. I absolutely agree with Definite. But when we can do this. And, I think that has forever changed the world of vaccinology, for me, at least. And I think the big, sort of challenge to the advocates on the call today, and I'm so delighted to be part of this, just great people who make things happen out there. The challenge for all of us is, how do we take that thinking and energy into the HIV and TB vaccine world and make it count? Thanks, Mitchell.
Linda Gale, thank you so much. And I'm so glad you put in TB there, because although we obviously have huge struggles in the HIV vaccine world, TB vaccines are, if HIV vaccines don't have enough resources, TB vaccines by an order of magnitude less don't have enough resources. So I'm delighted you brought that in. Next, let me turn to great friend and colleague, Matthew Rose, who directs U.S. policy and advocacy at HealthGap, a longtime AVAC partner. Matthew, and I should say a former vaccine trial participant. How does it all play out for you, and how has COVID vaccine R&D influenced you this year?
I think it highlights some things that we have been saying for a very long time in the HIV space. One is that it is so critically important to start building demand creation and be answering people's hesitancies about how a vaccine works and why a vaccine will improve their lives beyond the people who just want it immediately. And we have to start doing that work before we even have a product that works, because we have to start building that base level of understanding. You can watch it in countries as they kind of surge towards that 50% mark, and then are challenged getting further along than that, or things start slowing down. And that's what something that has a virus that has much more immediate impact, that is COVID. HIV has a much longer timeline. There's still a lot of stigma that we're gonna have to overcome. But I think that more people than ever are curious about, what does adult vaccination mean? What does it mean to talk about antibodies? What does it mean to have large scale systems who are rolling out vaccines in places, and figuring out some of the logistics for that and what you kind of have to put in place? I think we learned a lot about the importance of primary healthcare being a site for people's introduction and understanding of how a vaccine works and how people engage with it. And so I think we've gotten a lot of lessons, and we've also given a lot of lessons and a lot of sweat equity to helping the COVID process along. It's not lost on me the number of platforms that are used by COVID that have been drastically advanced or created or first utilized within the HIV space. And so I think there is some great cross learnings, and I think that we can take those to continue accelerating the pace of our technology.
Fantastic, Matthew. That was great. And our fifth panelist is Pantiano Kalibu, a longtime advocate and researcher in Uganda at the Medical Research Council UVRI program related to the London School of Hygiene and Tropical Medicine Ugandan research unit as well. Pantiano has led multiple vaccine trials in building infrastructure throughout not only Uganda, but throughout the continent. Pantiano, I'll turn it over to you to give a sense of the last year.
Thank you very much. I hope I'll be audible enough. My reception may not be very good, but thank you for the opportunity. I think you were asking how COVID vaccine research has influenced our HIV work and research. I think the first presentation from Daisy, I think clearly showed us how some of the way the funding, the coordination, the way we have worked together, but also the difference between HIV and COVID has allowed us to have the COVID vaccine quite quickly. It's been very exciting year. Exciting in that, in spite of the fact that, okay, there's more funding, coordination, but I think they have shown us that the science can work. The science is good. I'm very happy that we have used the old concepts, inactivated vaccines, but also the newer concepts, messenger RNA and all that to deliver a vaccine. To me, it has indicated, and I've been presenting it to colleagues, that the science and the scientists have been working. They're not hopeless, but it shows how difficult HIV is. HIV virus is different from COVID. It's one of the challenges that we have. Daisy clearly explained that those who have COVID, many of them recover. HIV, you never recover. So it's complicated. It's a virus that is different. But the scientists, I think, are doing a good job. They have been able, at least with the COVID, to show. And it's very good for the young people who have seen a pandemic in front of their eyes, science in front of their eyes, and then within a short time, see a vaccine, what science can deliver. It's so exciting to be a scientist at this time. In fact, in Africa now, we're talking about how can we contribute more so that we're not just bystanders. How can we contribute and design vaccines? There's a lot of pressure now because we have shown that science can work. So it's really the COVID experience we've had has been a boost to science. Invest in science. Science can really solve a number of our health problems. HIV is different from COVID, but at least the science has been fantastic to be able to deliver a vaccine within just 1 year, a very short time. So really, it's been exciting. To me, it's a boost to HIV research. And it's also clearly, it has given me a way of presenting that why HIV vaccine has been difficult. It's not necessarily that the scientists are hopeless, but we need to really invest in science, put in more money, collaborate, work together, and we can deliver. Thank you.
Thank you so much, Ponciano. And the scientists are far from hopeless. You're our heroes currently. So I think I will turn to Professor Barney Graham with a question. I mean, I think several of the panelists have mentioned the mRNA vaccine, that the COVID vaccine that a lot of people, especially in the Northern Hemisphere, are currently using. Dr. Graham was one of the pioneers in that work. And the question I would like to pose to you, Barney, is how does it feel? How was it to develop this mRNA vaccine? I'm sure it was a long journey. I mean, we're just seeing the results now. Thank you.
Yes, I loved what Ponciano said because my children have always teased me that every time I got a cold or they got a cold, they asked me, I thought you've been working on cold viruses and vaccines for 30 years. Why are you still getting colds? So it does feel good to have the technology available now to make a vaccine that can work even against the common cold, and in this case, much more serious than the common cold. But mRNA is a technology that I have great hope for, and it's I think people have seen its potential this year. It's the simplest possible way of delivering a protein vaccine antigen. And it's very simple. It doesn't last long in the cell. It goes in, it does its job of making protein, and it's gone. And so I think you're seeing the benefits of that type of vaccine antigen delivery through a nucleic acid. But my great hope for this is that because it's a new way of doing chemical synthesis for manufacturing, it creates the possibility of bypassing some of the big bulky equipment needs for conventional manufacturing with bioreactors, where you almost need a whole campus of buildings and equipment to make a vaccine. mRNA gives the hope that we could potentially have small manufacturing footprint capability that could be better distributed to low and middle income countries, where even modular GMP units could be used to make small batch manufacturing lots for regional problems and regional diseases before they become pandemic threats. We can say more about it later, but maybe I should just add too that mRNA is a beautiful, elegant way of delivering a vaccine, but if you don't deliver the right thing, it's not going to matter. And so people have to remember that this is a way of delivering a vaccine antigen, but if the antigen isn't the right one, or isn't in the right conformation, or doesn't have the right properties, it won't elicit the right immune response. And so it really has to be combined with antigen design to make the vaccine work.
That's really great. A synthesis of the mRNA platform, Dr. Graham. So we've been hearing about the possibility of using this technology to make HIV vaccines. And we were just wondering if you could give us your quick take on how realistic this is, and how, feasible, and, can it be done?
Well, Daisy, you explained all the biological challenges there are in making an HIV vaccine. And to date, we don't really have vaccines for viruses that have, not very good ones anyway, for viruses that have this type of extreme antigenic variation or antigenic diversity. And we have an influenza vaccine, but it's not that good, and it really only works against matched strains. And so the problem of antigenic diversity and a related phenomenon that I call immunodominance, that elicits immune responses to parts of the virus that don't really hurt the virus. And so the problem of antigenic variation, the problem of immunodominance, and the problem of having a virus, as Pontiano said, that is not naturally cleared by the body, and so it becomes a persistent infection. We don't really have very good vaccine examples of things that can prevent and clear viruses with the capacity for persistence. So these biological hurdles are gradually being solved. I think people through structural biology have solved one of the big problems of conformational evasion, where you just don't have the protein in the right conformation. Well, now through the work of many groups, that envelope conformation can be preserved, but it's still blocked by the glycan shield, and there's these other biological, immunological problems that make HIV still a difficult problem that we are still working on. So mRNA is not going to solve those problems. mRNA is a beautiful way of delivering a protein, but we have to make the right protein.
It's incredibly helpful, Barney, because I know we had a number of questions that came in before the webinar when people registered asking about mRNA application to HIV, and super helpful to see that. Maybe I'll turn. One of the other big platforms, of course, is the adeno-based vaccine. And Linda, you've been involved in both the Ad26 trials for HIV vaccines, as well as the Ad26 COVID vaccine program in South Africa. But I wonder particularly, there's a great question from Pumzile Suguntu asking about access. And I wonder if you might spend a minute talking about how you and colleagues in South Africa have begun to transition from clinical trials of the COVID Ad26 into the access program of Sisonke, and what maybe lessons we might have going forward about doing better to accelerate access.
Yeah, Mitchell, perhaps the one thing we haven't learned well from the HIV pandemic to the COVID pandemic is just this quandary of the haves and the have-nots, right? That the world remains an incredibly inequitable place. And, at this moment, pretty much every country in Africa has less than 1% of its population vaccinated. And in a few countries where we are seeing vaccines, we certainly aren't seeing what might be considered, the superior vaccines. And I'd certainly put the mRNA vaccines into that category in terms of, side effect and just impact and vaccine efficacy. Having said that, we were absolutely delighted to be able to participate in the Ensemble trial, the AD26 COVID vaccine. And again, I want to underpin and underscore the importance of running these clinical trials on this continent, because I truly do believe that it opens doors for advocacy, for, if you like, creating the space for the conversations to say, what about us, and how do we get these products to us here? I truly think it's because we had Ensemble here in South Africa that when we hit the unfortunate hiatus with no vaccines in country, or prospects at that point for vaccines for a long time in South Africa, we were able to reach out and ask if there was any possibility of conducting a extended phase 3B trial, as a way to lay our hands on any vaccine at all and be able to safeguard the healthcare workforce in the face of a third wave. And I will tell you, I'm not sure yet if this is the third wave, but today, just about every single province is showing an uptick in the 7-day rolling average of new cases. And certainly around the country, it does look like new cases are taking off. So not a moment too soon that we have been able to start the Sisonke trial. We started on the 17th of February. We actually wrote the protocol a week or two before that. We got the regulatory, and this is what I mean by, rapid fire activities. We were able to get started on the 17th of February. And as of this evening, I expect to see about 480,000 healthcare workers vaccinated with the AD26 vaccine in the last, and this all through a research team. So as you all know, our Ensemble team is probably about 35 research teams in country in South Africa. They have all partnered with vaccine centers, and single-handedly, we've kind of churned through these almost now 500,000 healthcare workers. Without doubt, access is very closely linked to R&D and to the process of bringing new products to countries. I think the challenge that sits ahead of us of how we, in the future, make sure that when new products, new commodities come and they have public good the way a COVID vaccine does, that we have some way of making sure that we can get them to all the corners of the world where they're needed. I think, Mitchell, it again very much underscores for me the importance of research going hand in glove with the question of access.
Fantastic and incredibly exciting to see that linkage that you're beginning to build. There was one question that did come in from the registrations, maybe particularly for you, Linda Gill, just to pick up on any information about people who might get ADNO26 vaccines for both COVID and for HIV. Anything that people should be aware of? Is there any reason to be concerned about multiple ADNO vaccines?
Yeah, I think where this is coming from, Mitchell, is that clearly when a viral vector is used as unlike the mRNA platform, the viral vector platform as a way to deliver the vaccine, the host may develop immunity or a resistance, if you like, against that particular viral vector. So we certainly saw that with AD5. And it has raised some questions about the AD5 vector because we see a lot of background immunity to AD5. We're a continent where we pass infectious particles around a lot here. And so we saw very high rates of background immunity to AD5, and it's thought that that could undermine the value of the vaccine. Many years ago, Dan Baruch and a number of us looked at the rates of background immunity for AD26 and happily much lower than AD5, but there remains some concern about that. So again, the theoretical worry is that if you repeatedly go in with the viral vector as your platform, that you might increase the background immunity to the viral vector and then undermine the vaccine. I think we haven't had enough experience to know if that in fact is the truth, but I think it remains a theoretical concern. Having said that, there are some values in using a platform for more than one vaccine. So, we were able to, with impunity, talk about the safety of the AD26 ahead of time, because we've obviously also used it in Zika, in Ebola, and a number of different other situations. We do have, and I'm sure everybody's very aware, we have had some safety risks and concerns that have come up around the AD26 and the chimp AD vaccines in the COVID space. But again, we have to say we haven't seen those thrombogenic abnormalities in other vaccine trials, which I think does raise what some of the mechanisms might be for the difficulties we're seeing with the COVID vaccines. And I think this remains an area for research. But, I think there is that value of when you have some comfort with a particular medium, to be able to use that in more than one vaccine has some spillover good effects as well. So I think, pros and cons to that.
Thank you, Professor Linda Gale. I think we're going to pivot to the resources that are needed. And Jim Pickett put a question on the chat, on the Q&A, regarding what it would take to really go big with HIV vaccine research. And I think I'll pose that question to Matthew, because Matthew is an advocate that has been in the policy and advocacy space for a long time. Matthew, what would you say it would take for us to really go big in the push for an HIV vaccine in terms of policy and advocacy?
Yeah, I think that it goes a couple layers, and I think that there's not resources has to be thought of kind of in a broader context as well. I think that COVID has kind of laid bare that we need to have better manufacturing and research capabilities to design vaccines in other places. I think being able to kind of expand Barney's work. I know every year we have these like weird discussions about what we're going to do about HIV research. But we only have funding really in some places to only do a couple big trials. And we don't have the ability to do kind of some more complex trial designs that might be more advantageous to finding a vaccine, but are going to be larger, especially as new prevention products come online. So there's just going to need to be more funding in that space, but also just the human capital of having talented virologists and vaccinologists see HIV as a space where they can continue doing their work and want to come over to do our work. I know there's definitely an appetite, and people have definitely made a lot of illusions over COVID about what HIV research has brought to the table. Our research networks were critical in getting the Moderna vaccine tested and some of the Pfizer work done, or not some of the Pfizer, excuse me, some of the J&J work done. So clearly we have platforms that are helpful, and expanding on those, we can show kind of what we're able to do. But it is a multifaceted piece about being able to have the money to do it and then being able to spend those in ways that do new things, like potentially having more adaptive trial designs, available. But there are some key considerations, and some of that is people making the demands on their governments to be supporting this type of research and showing that this type of research not only teaches a lot of things about HIV and hopefully gets us to an HIV vaccine, but also spillover effects in other areas as well.
Thanks so much, Matthew. And you know, I'm so glad you framed it that way, because so often we think about it being about dollars or rands or euros, and you're so right about the resources being more than just financial. And to Jim's question, you know, resource tracking data show that, you know, it's been relatively consistent, about little over $800 million a year for the last couple of years. It's not so much how much more money, although more money is always helpful. It's how it gets spent, and then the leveraged resources beyond the money itself. And I want to maybe turn then also to you, Def. You know, one of the big issues that's come up, and it's in some of the questions and certainly some of the conversations the last few weeks and months, have been about confidence in both the research process as well as in products themselves. And you know, you've been a great advocate for both research, also for the rolling out of PrEP, and now looking towards long-acting injectable PrEP. And just wondering if you could spend a minute or two talking about what should be done differently as we think about building confidence in both the research process, but also in the products themselves.
Thank you so much, Mitchell, and great conversation here. And I think in terms of confidence in the product and the research process, vaccine hesitancy has been one of the biggest issues we are facing with the COVID vaccine. And I think we should do more if we have vaccine hesitancy for COVID, thinking ahead, what will happen when we have an HIV vaccine? Do communities still have that confidence in research, in the research that's happening? Do they have confidence in the licensure period, in the registration periods? What happened in that pipeline from research to access? Will people access the good products that we are spending all this time investing into? What should we do? I think as an advocate, what we need to do is community involvement is really important, because whatever product researchers are developing, product developers are developing, researchers are researching on, it needs the end user. So if the end user is not on board, then we'll have products that are on the shelves, but then no one is taking them up. Taking COVID as a great example, for example, in Zimbabwe, we have just under 3% of the population being vaccinated. The big question is, when do we get to reach that herd immunity? A lot of people have questions around the COVID vaccine. Is this the real vaccine? Is this the real thing, or it's not? For example, for Zimbabwe, our vaccines have been donated. And then people are wondering, if there's no political will to buy these vaccines and we get donations, should I go for this vaccine, or should I not go for the vaccine? And also another question is around, because we're hearing of so many mutations around even COVID, which also happens in HIV. How many boosters does one need after the initial shot? Is it just the first and second one, or there are so many? And in terms of cost, how sustainable is it? Are people expected to then pay at some point? What is going to happen? And another issue that I really want to bring up is the issue of, we did hear about the few cases of blood clotting after the COVID vaccine, and just trying to understand whether this has also happened in the HIV vaccine world. If it has, what has happened? What mitigatory measures have been put in place? And but the last issue I would really want to highlight is the issue of trial designs. Moving forward, with the vaccine research. What is it going to look like when PrEP is the standard of care? Are we going to have enough people? Are we going to see enough new HIV infections for us to get the results that we desire in terms of the HIV vaccine? So these are all questions coming from me as an advocate, and would really love to hear some of these responses, time permitting. I'll stop here for now. Thank you so much.
Jeff, thanks so much for your observations, and I'm so glad you mentioned trial design, because I would like to turn to Professor Pontiano, who is heading up one of the most interesting vaccine trials. There's a vaccine trial happening in 4 countries in Africa: Uganda, Tanzania, Mozambique, and South Africa. And this trial called PrEPVAC was actually started in the middle of the pandemic, I think in 2020, just as the pandemic was starting to really disrupt everything. So Prof, could you please share your experience about how did you do it? How did you launch PrEPVAC amidst all that?
Yes, thank you for that. Indeed, it's not been easy. Before the Rio trial, which started, at least we started enrollment into the trial end of last year in December here in Uganda, we had started with what we call a registration cohort. And with this registration cohort in the different countries and sites, we're enrolling individuals who could potentially also enroll into the trial. First of all, to study these populations, to estimate the incidence in this population, but also to understand them, acceptability and all that. That did help. And of course, unfortunately, the registration cohort, as we were following them, was very much interrupted by COVID, but we had very good follow-up. And we could also, that registration cohort could indicate to us which sites were potential to enroll into the trial in terms of their retention, in terms of their incidence, in terms of their population characteristics. So that was quite helpful. So the registration cohort then was followed by the trial itself, which is a PrEPVAC trial. It is comparing different combinations of vaccines against a placebo, but also within the study itself, we also have a study looking at PrEP, comparing Truvada with the Descovy. So it was, most of the COVID did disrupt definitely, and we had to pause and in many of the sites, but also our staff, some of them got infected with the COVID and they had to go into isolation. But now, luckily so far, things have stabilized a bit with less new COVID infections, and the trial is ongoing. Here in Uganda, we have already recruited 81 participants into the Rio trial, and the other sites in Tanzania, in South Africa, in Mozambique are also preparing to start recruitment into this efficacy phase 2B trial. Yeah. Thank you.
Great. Thanks, Pontiano. And I think all of you on the line are involved in various activities that happened in the midst of COVID. It's quite remarkable. PrEPVAC got up and running. We've seen results from multiple HIV trials. And I want to turn to one of those and related to one of the most recent results, and that's the antibody-mediated prevention trial using an antibody actually named after Barney's organization, the Vaccine Research Center, VRC01. When that trial was first designed and discussed, it was very much the thinking that antibody research would influence vaccine development and not necessarily be a product of its own if one goes back a decade or so. But things have changed a lot. As Daisy said, antibodies now get talked about in the COVID landscape. But I wonder, and maybe asking each of you really quickly, just to provide a perspective on the AMP results, which, as Daisy said, were very complex. And I will just put a plug in for our most recent podcast that Daisy and a colleague Jean Barron did with researchers and advocates looking at the AMP results. But just wonder if, and maybe we'll start with you, Barney, what does the results coming from AMP mean potentially for vaccine design?
Thank you, Mitchell. Yes, I know the results have been a little bit hard for some people to understand or interpret, because overall there was no overall significant efficacy with that product given in that way. But to us, we thought it was a real breakthrough study, because it gave because of the work on the breakthrough infections and understanding exactly what level of antibody and what level of neutralizing activity was in serum at the time of infection. It gave us a way to calculate what level of antibody function will be needed to prevent an infection. And so for us, it was sort of a breakthrough result. And we found that antibodies at that level that had neutralizing activity of greater than an IC80 or an inhibitory concentration of 80% at 1 microgram per mil concentration, that is going to prevent infection with that virus. And likewise, neutralizing antibody level in serum against a particular virus of around almost 300 is sufficient to protect against that particular virus. So it gives us a benchmark that we can try to target with both vaccines and new antibody combinations that we think if we can hit that mark that they should work. So we thought the trial was very important for that reason.
Great. I'm happy to see others, Matt, LGB, Pantiano, Def. Any thoughts on the AMP results and what they might mean and influence for the future?
Mitchell, I'll jump in. I'll absolutely echo what Barney's saying. For me, it was an amazing sort of ray of hope. I think it opens two avenues of inquiry in my sort of simple clinical trialist mind. The one is the kind of, I suppose you put it more into the pre-exposure prophylaxis bucket. I like to think of it as passive immunization, where, you know, we now have a wonderful pipeline of very exciting antibodies, and could combos be put together, much like we saw with COVID, in terms of using antibodies either to bridge the time to immunization or to use in very explicit and important places, for example, in the prevention of mother to child, for instance. So I think there's a whole field of investigation that needs to happen there. And then secondly, sort of more complicated, as I say, for my more simple mind, but I'm very inspired by our vaccinologist science friends, people at IRV, people at the VRC, and others who say, you know, we can back engineer what a vaccine might look like using this knowledge, knowing that we can in fact stop HIV if we get the right configuration of antibody at the right place. So I'm incredibly enthused and excited. I think it does open you know, the next era in HIV vaccine research. And I just, you know, my last plug is for clinical trialists. I think the AMP study was an extraordinary study. At the beginning, I have to say, I'm really rendered skeptical, but right at the beginning of the AMP study, I kind of scratched my head and wondered how we were going to do this. And, you know, when you read how beautifully that study was executed and conducted, and the incredible commitment of people on this continent and other continents around the world who took part and enrolled and showed up for that trial, I have huge hope for what we can actually achieve in the next few decades. And I'm looking forward to, you know, what is to come. Thanks.
Yeah, just to jump
Yeah,
in, Mitchell,
yeah.
just 1 minute. I think for me, the AMP study really opened a whole new world of possibilities, but humbly showed us that HIV virus is it's a tricky virus, and there's still a lot to learn in terms of on antibodies, and just wondering how and when we are going to have that breakthrough, but really hopeful, just encouraging researchers to work together with communities, advocates, so that we walk together down this road. We understand it's not an easy road, and we are all trying to figure it out, but let's work together, let's work together in this. Thank you.
Just for me, a brief comment. Again, to me, that's a triumph for science. I think when you look at where we started, and again, I think the contribution of Barney and VRC and many others are having into really identifying these antibodies and being able to make them. It was a proof of concept. And I think the results we got, to me, they were exciting, not surprising, because as the trial was being conducted, there was, of course, that indication that maybe one antibody alone will not do it, but at least it was able to show some effectiveness for viruses that are neutralizable and for other reasons. And what Bana has mentioned, the assay itself, showing that at least it can indicate to us which are the titers that could be protective. But I think it is a big advance for science to show that these antibodies, again, neutralizing antibodies, could block. This is where we started with drugs, with a single drug, AZT, but it seems you need a combination of these antibodies. I think now the other area that will need to be worked on is a better delivery. Of course, we can't continue with the infusion that was used. Better delivery, I think, will be key. But I think the AMP trial and all the work on this antibody is really very important for science to prove the concept that the antibodies can work when you produce them. But as it has been said, the virus is complicated, but I think it's a big advance in science. Thank you.
Yeah, thank you, Prof, for your remarks. And as we know, there are some combination antibody trials that are just about to start. So we look forward to those. So we are at the top of the hour. Actually, we are a few minutes past, but I will not, I think Mitchell and I wanted to end it with coming back to the HVAD theme. And I would like to ask each one of you to give a 30-second or less HVAD final thought. I will start with Matthew, please. Matthew Rose.
Oh, I was giving credit to let some of the others go first.
Oh, no, you go first.
HVAD is always a wonderful day to think about how close we are getting and where we're at and how we shrink the window. And every year I think about it, like, have we put another piece into the puzzle, knowing that there's not going to be a silver bullet that gets us there, but it is going to be in a true way of science, is that we've got to advance, iterate, reapply, and stretch ourselves. And every year is a reminder of what the power of vaccines can do, especially in this era of COVID. And so it is particularly important to kind of honor, acknowledge where science has taken us, but where the community pieces continue to fit in, about people knowing what's going on, people being aware. The day is literally about awareness and understanding that an HIV vaccine is potentially within our grasp. We keep getting these key notes that tell us that we're closing the gap from where we need to be to where we can get. And I'm just excited for the way and path forward.
Thank you, Matthew. Let's turn to Def, please.
Thank you, Daisy. For me, just to say these are exciting times. I think the COVID vaccine is really exciting. It's something we did not anticipate, but it did show us that things can be changed. Things do not always need to happen in the way they're written out to, but things can be changed. We can change the trajectory. And we're really looking forward to changes in the HIV vaccine world. We're looking forward to being surprised one of these days in terms of the HIV vaccine. But these are really exciting times. And thank you so much for the engaging conversation today.
That's great. Thank you. Prof. LGB, we'll go to you.
Thanks, Daisy, and thanks, Mitchell, for this opportunity. What I would say is such an exciting time to be alive for prevention in HIV. And if you haven't read about it, you need to go to the AVAC website and just see the incredible breakthroughs we've had in the last 10, 15 years in terms of HIV prevention. But it does not for a minute diminish my contention that the world needs a safe and efficacious HIV vaccine as soon as possible. And I'm right there behind all of the advocates, pushing for this. We really need to keep our commitment high. Thanks.
Thank you. We can hear from Professor Barney Graham.
Yes, I remember that we sometimes celebrated HIV Vaccine Awareness Day when I was at Vanderbilt by planting a tree. And for me, vaccines represent hope and hope for the future. And my hope is that with some of what's happened this year with COVID, that it will turn back to help us advance our ability to prevent HIV infection. And the hope for the future is the next generation of scientists who are going to have technologies that can, I think, will give us unimaginable results over the next 20 years. So I still do have hope that we can find an HIV vaccine.
Thank you. And last but not least, Professor Kalebu, before we turn it over to Mitchell to close out the webinar. Prof.
Yes, I think the HIV Vaccine Day comes at an exciting time, especially for the COVID, but also for the many other HIV prevention approaches that have come on. But I think using the COVID example, it further shows us that we need to work together. Everyone is important. The community is important, advocates are important, the funders, the science, everything is important. We now have a COVID vaccine, but there's the issues of access. Not everyone can have the COVID vaccine. There's hesitancy. There are people who are anti-vaccine, who are talking louder than us. So it really shows us that if we have very good vaccine, good science, there are other areas that we need to address. So that, because what we need is the vaccine to reach the people who need it. So I think let's emphasize again as we celebrate the successes that indeed everyone is important in order to really fight this HIV. Thank you.
I can't think of a better set of comments to reflect on Vaccine Awareness Day this year and always. The richness of what all of you said and the multiple stakeholders. And I think something, Pontiana, you said at the beginning of the call, Matt, you said it toward the end, the sense, and everyone in between, the sense of excitement and enthusiasm in the science, but also the sense of challenge of making sure that those scientific advances actually have the ultimate public health impact for COVID, for HIV, for TB, across the board. And so I always look to May 18th as a day to be reminded of what is possible, the enormous amount of work done, and the enormous amount of work still to come, and that work to be done collaboratively. And it's really just a day for me to be reminded of colleagues, partners across the board who advanced this work. Very importantly, one of the important aspects of Vaccine Awareness Day too was to acknowledge the participants in research. for all the great science, for all the great vaccine platforms, for all the great advocates and researchers. Without trial participants, we will never get new innovation, vaccine and otherwise. So I want to acknowledge that and thank everybody for being on the call. And perhaps more than ever, 24 years later of Vaccine Awareness Days, I think for us at AVAC, it is increasingly both about being HIV aware, not just vaccine aware, because we have this growing range of options that we now need to deliver as choices to people: oral PrEP, vaginal ring, injectable cabotegravir, the next generation of products to come, including vaccines and antibodies. It's an exciting time and an important time to be HIV aware. And it's an critically important time to be vaccine aware, and not just HIV vaccine aware. So perhaps we'll rethink about what we call this day, but the commitment to science, to equity, and to collaboration is one that no matter what we call the day is something we should be doing every day, and look forward to that in all of our work going forward. Daisy, thank you always as my colleague and partner, but Linda, Gail, Barney, Def, Pantiano, and Matt, and all of you who joined. We have a lot to do, but I think we have a much stronger platform on which to do it. So thank you all. Stay safe and well and engaged and aware, and appreciate everybody's time.
Thank you very much. Thank you. Thank you.
Thank you, everyone.
Thanks. Thank you. Take care.
Nice to see
Thanks.
everybody.
Bye.
Cheers all.
Cheers.